So here's lesson 3, water and macromolecules. The first thing that we'll do in this lesson is we will talk about why water is essential to life. We will talk about a few basic properties that you might already know about. And then the last one, chemical reactions, is where we'll go into a bit more detail because this is what we're going to discuss in this biochemistry section. So I'd like to remind you how important water is to life. When we do the last section of biology, we'll come back to these properties of water again. One of the properties of water is that it's transparent. It's see-through. And so I made a little table where I put some properties, and on the right, I wrote significance to life. Why would it be important for a living organism that water is transparent? The second property, I actually combined a few things there. The word I used is polarity. If you never learned or you forgot what polarity means, polarity, or water being a polar molecule, means that it has a positive and a negative side. And because water is polar, it has these other properties. Because water is polar, it is a good solvent. Things dissolve in water. And because water is polar, it displays cohesion and adhesion. Now, I'm sure you heard those words, Science 10, biology, plants, when you talked about xylem and phloem and how water moves through a plant. The third thing is heat capacity. Comes from Science 10 as well, the last unit, where you did Q equals MC delta T. Maybe your teacher played you a lame rap of a guy who was MC delta T, and he sang a stupid little song. But water has a high heat capacity. That means it stores heat very well. For at least the first three, since I think you're familiar with them to some degree, I'm going to stop talking for a second. So we'll start with transparency. I saw at least a few people had an idea of why water being transparent would be significant to life. Can someone share with me what they wrote? So, over here, I heard you say it, so can you say what you told me? So birds can catch fish. If water wasn't transparent, would organisms be able to see through it and be able to live in it and see what they were doing? So I'm going to, oh, that was funny. I'm going to write vision just in general. If water wasn't transparent, organisms that rely on vision would not be able to exist around water or in water. Did anyone think, oh, you have one, yes. I see where she's going. If it was not transparent, light would not be able to pass through it, and plants would not be able to perform photosynthesis. And I'm going to point this one out specifically because we're going to talk about photosynthesis, very next thing after biochemistry. Let's talk about polarity. I saw a few people write something down for polarity, maybe because you remembered it. Does anyone want to volunteer their answer for polarity? Is that your hand, Grace, or no? No? Luis, I saw you had something. Do you want to share what you wrote? It's right. I looked at it. So, in a plant, these forces, cohesion and adhesion specifically, they're the idea that water sticks to things. Cohesion is that water sticks to other water molecules, and adhesion is the idea that water sticks to other molecules. And so water can be brought up in plants effortlessly, like you said, because of these forces. It prevents water from falling back down, but instead forces water to continuously move upwards from the root all the way to the leaf. Did anyone maybe think about how water is a good solvent? So I'm going to ask you, what are you made of? If I asked you what's the number one thing that you are made of? Anybody know? Yes. It is water. That is an excellent guess. So you are mostly made of water. How about blood and the fact that it dissolves stuff, a whole bunch of stuff? We will talk about your blood a lot in our unit on human physiology, not just when we talk about circulation, but in fact when we talk about every other body system. Without blood, nothing can be transported in your body, because blood is the transport mechanism. Let's do heat capacity next. Did anyone have something there? You did? So we're talking about like state changes, I think is what we're getting at here. So because water has a high heat capacity, it's relatively difficult for water to change state, and when it does, a lot of energy can be released. So something that you might remember hearing about is that when water changes state, there's a specific amount of energy that's required to change, say, water from a liquid to a solid. The result is that when water changes state, it can help warm up the environment around it or perhaps cool off the environment around it. The details of that are a Science 10 or a Chem 30 topic, so I'm not going to ask you to calculate this, but just to recognize that water changing state can affect the environment around it. The last one is chemical reactions, and you likely have not heard of these two things before, but the two reactions that we are going to discuss are dehydration synthesis and hydrolysis. These are basic metabolic reactions, so they're part of the reactions of metabolism, that occur all the time in living organisms. They're the way that we make macromolecules and that we break them down. So we're going to talk about each of those reactions in turn. Before I move along, does anyone want to ask a question here? So first, we'll talk about hydrolysis. Hydrolysis kind of goes hand in hand with digestion. So we'll mention hydrolysis again when we're talking about digestion. But when you're trying to digest food, carbohydrates, proteins, or fats, the macromolecules, we need to break, what happened to my pen? We need to break the chemical bonds that hold them together. And hydrolysis, like a lot of science words, has two parts. Hydro and lysis. Does anyone know what either of those two small word parts means? Yes, William. Hydro definitely means water. Does anyone know what lysis means? I'm going to go with the simplest explanation. It cuts, breaks, tears apart. So lysis is when something is being taken apart. I like to use cut because it gives a better visual. It's like a pair of scissors cutting or snipping something off. But that's what lysis is. So hydrolysis is literally water cutting a molecule apart. What happens, if you see here, you add a water molecule into the structure of a carbohydrate or a protein or a fat. And by doing that, water shoving itself into the molecule ends up splitting the molecule in two. So, I'm going to give you a little analogy here. Have you played Red Rover? I did. I know my biology class last year, there was a girl in it who was quite proud that she broke someone's arm playing Red Rover. I said, are you sure you want to be proud about that? It's a little harsh. But in Red Rover, the idea is that you're holding hands and you look at the other team and you say, Red Rover, we call Grayson over, and he comes running full speed and tries to break the spot right between us, so tries to make our hands come apart. In that analogy, Grayson is water. Me and my friend who are holding hands, we are the carbohydrate. And if he gets through, if he wins, Red Rover, he inserts himself into our team, right? He puts himself into our team at that spot, and he has essentially broken the bond between me and my friend. We are no longer holding hands. That's what water does. Water meets up with a macromolecule, any type, carbohydrate, protein, or fat, inserts itself, and then splits the compound into two smaller compounds. So on the right, I have a picture. It is an actual structure. I would not ask you to memorize in full detail the actual structure of a carbohydrate. I just like to be specific. On the bottom, I have a large carbohydrate, and on the top, I have two small ones. And you'll see that I'm using this picture on this slide and the next one because it represents both processes. If I take my large carbohydrate that's on the bottom, and I add H2O, and H2O will add itself right in the middle there, right in that space between the two hexagons. The result is that it will be split in half in the end. I will not ask you to recreate a picture. I might ask you, hey, here's a picture of it happening. What is this? But I will not ask you to draw from scratch a detailed diagram that shows me the exact molecule where it's happening. Does anyone want to ask any questions there? Okay, so we really want to focus on the fact that I added water. If you like visuals, I'll just show you. See how in the middle there was one O? And now in the middle there's two O's and two H's. It's like I added H2O right in the middle. So I literally took those atoms and attached them. One to this side, a couple to the other side. And that's what happened. Now we'll use the same picture, so it's this exact same diagram, to explain dehydration synthesis. Dehydration synthesis is the opposite process. So I always like to highlight when things kind of go hand in hand. Whenever you have two things that are essentially the opposite, that immediately makes a good test question, because I can ask you, is it this or is it that? This process involves synthesis. The word synthesis is where I'm putting things together. That's what it means, to synthesize something. And if I look at the other word, dehydration, I'm unwatering it, so I'm taking the water away. So the opposite process is to take my two small molecules. So again, I have the two small molecules up here, and I will remove a hydrogen atom and an OH group. So I'll just highlight them. Hydrogen, OH. So I need to remove two H's and an O, and they can't all come from the same side. One H has to come from one molecule. The O and the other H have to come from the other one. Now the fact that I pick left and right, I could have reversed that. I could have said the O and the H come from the left one and the H comes from the right. That's irrelevant in this particular thing. But I remove those things. They join together. So that's where I get my dehydration synthesis. I could also use the word condensation. If you're going to pursue chemistry, in Chemistry 30, when you do organic chemistry, they like to use the term condensation for this type of reaction. Both processes require enzymes to make them faster. Now, enzymes are something we will discuss in a lot of detail, and we'll do a little activity about enzymes. Enzymes are molecules that make reactions faster. For now, that's all we need to know. So, carbohydrates, they're one of the types of macromolecules. You'll see I put CHO, which is the same thing I put for macromolecules, because that's generally all that carbohydrates are made out of. If I look at the word carbohydrate, what does it sound like? Kind of sounds like carbon and water. And that's what they're made out of. They're made out of carbon and essentially molecules of water. Of course, things are rearranged, but that's a carbohydrate. So it's made out of C, H, and O. The purpose is something that you'll need to know for all the types of macromolecules. What is their function, their objective? So the purpose of a carbohydrate is energy. Now we'll talk about energy for fats as well. And so for carbohydrates, I'm going to add the word immediate. Carbohydrates are your source of energy right away, right now. It's easy to access. Now, carbohydrates aren't just for energy. They also are structural components of cells and cell membranes. If you learned about the cell membrane, you might recall hearing that the cell membrane of a cell has phosphates and lipids in it. It also has proteins and then sometimes carbohydrates sticking off the end of it or the side of it. But the main function of a carbohydrate is energy. Its structure, well, it's made of CH and O, and then I'd like to be a little visual with the structure because I think trying to say it with words is a little clunky. Over here, I've got a picture, and I'm just going to highlight the big idea. Carbohydrates usually have a hexagon shape as their base. Sometimes it's a pentagon, like in this one, depending on which sugar it actually is. But if you see a picture with a hexagon or a pentagon shape in it, what that actually is a line diagram in organic chemistry, and each corner represents a carbon, but that's not the point of our course. So I'm just telling you visually that's how we would draw a carbohydrate. Now you can have more than one type of carbohydrate, a mono, a di, or a polysaccharide. I told you what this word meant the other day, but I'll repeat myself. Mono means one, di means two, poly means many, and the saccharide portion of the word means sugar. So I could have carbohydrates that are made of 1 unit of sugar. A monosaccharide would look something like that first picture, one hexagon-looking shape. A disaccharide would be something like the second picture, a hexagon attached to a pentagon, or a hexagon attached to a hexagon. And a polysaccharide would be something like this last picture, where I have several hexagons attached to each other. Notice I'm really glossing over the detailed structure of each of these because I don't need you to memorize chemical structures like that. I need you to be cognizant of their approximate shape and what elements are part of them. Carbon, hydrogen, oxygen. Now there are 3 monosaccharides that I'll tell you about. Glucose, which lots of you would be familiar with because that is what you make in photosynthesis and what you use in cellular respiration. Fructose, which is a very similar sugar. It has the same molecular formula, but it's arranged slightly differently. And galactose. They are all 1 unit of sugar. Disaccharides all have this formula, so they're essentially double the size. They have twice as many carbons. Sucrose, maltose, and lactose We use 2 of those in the lab, you might recall. Sucrose is sugar, like white sugar that you would use. Maltose was sort of an orangey colored sugar you might remember seeing. Lactose is what's in milk. I want you to notice something. They all end with OSE. Isn't that convenient? Carbohydrates, especially the small ones, tend to end with OSE. So if you were worried like, oh, what is this? And you see that it ends with OSE, even if it was a new substance, you could probably guess and be fairly certain that it's going to be a carbohydrate. In the polysaccharides, there are 3 of them there. Unfortunately, only one of them ends with OSE. The other 2 have slightly different names. But polysaccharides are things that are made of many, and they're usually for more long-term storage or perhaps structure. Cellulose is found in plant cell walls. Starch is for plant energy storage, and glycogen is how you store energy in your muscles. So humans and animals would have glycogen, you have that in your muscles, whereas a plant would have starch and a plant would have cellulose. Does anyone want to ask any questions about the structure or the examples of carbohydrates? So then I'm going to give you one more bit of information, and it's specific to the disaccharides, because I'm going to show you how they're made up. Is there anybody who is still writing? I'll just wait for a second. So for these disaccharides, the next slide is going to tell you how they're made up. And I'd like to remind you that the way I would make them is through dehydration synthesis. Whoops, back to my pen. Right here. So dehydration synthesis, what we did on the previous slide, applies anytime I'm making a disaccharide or a polysaccharide. So of the disaccharides, we'll start with sucrose, because that's what the picture is of. Sucrose is this molecule down here. So you can see that sucrose has a hexagon unit and a pentagon unit, and they're attached together. Well, sucrose is made from, up here, glucose and fructose. What would I do to glucose and fructose? Well, I would do dehydration synthesis. I would take an H from one, an OH from the other, so that makes H2O for water. I would remove them, and now my 2 molecules can join up and become one. So sucrose is made from glucose and fructose. I like to make a little mnemonic here, something that will help me remember which disaccharide is made from which monosaccharide. So I took the first letters, S for sucrose, G for glucose, F for fructose. And my mnemonic is sugar makes you go fast, generally, and that's SGF. Lactose is another disaccharide. It's the one that's in milk. It is made from glucose and galactose. And then maltose is simply made from 2 glucoses. So when I look at this list, I can see that glucose is part of all of the disaccharides. So when I study it, I'm going to say, okay, I'm going to remember, like that's going to be something that's just going to be a given. Glucose is always there. And I'm going to focus on the other one, because that's the one I need to remember. That's the one that's different for all of the disaccharides. So you need to the way that works for you. That was Weird and creepy. You need to find a way that works for you to remember that fructose is part of sucrose, that galactose is part of lactose, and that glucose is part of maltose. I know for galactose, it kind of sounds like lactose, so I find that one fairly easy. But a mnemonic, maybe some sort of sentence that you make up, any of those types of things to help you remember this. I'm only going to ask you one question about this, hey? So I'm not going to ask you to recreate the whole thing. But I might ask you, here's sucrose, which are the monosaccharides that make it up? That would be something that I would ask. Was that your question? Yeah? Okay. Does anyone want to ask anything else about disaccharides? Okay, then last slide for today is about lipids. Now, lipids is the general word, and then you'll see I put fat and oil. And very often we use lipid as, or the word fat as an interchangeable word for lipids. Fats are generally solid, oils are generally liquid, and lipids are any of those things. Now you'll see after the word lipids, I wrote CHOP. For carbohydrates, I just had CHO. For lipids, I have CHOP. And the fourth element is phosphorus. And that's because most of the lipids that we encounter are actually phospholipids. So what's the purpose? Well, the purpose is to store energy, and I'm going to add long-term. Carbohydrates are for immediate energy, energy that you could access easily. Lipids are for long-term energy storage. But they do other things. They protect your internal organs. A bunch of hormones require fat or lipids, and they are structural components of cells. So lipids have a whole bunch of other functions as well. Underneath, I distinguish a little bit more between fats and oils. Fats in general, I mean, this is not a hard, fast rule, but fats in general are produced by animals, and they're solid, whereas oils in general come from plants and sometimes animals, depending on what the animal is, but they are generally liquid. So those are the most of the time. Most of the time, this is where they come from and what state they're in. Now, there's really two types of fats. There's triglyceride fats and phospholipids that I'll mention. We'll focus mostly on the triglyceride fats because you already talked about phospholipids in grade 10 and anytime you ever talked about a cell membrane. A triglyceride fat, well, if we look at the word, tri means 3, and glyceride has to do with glycerol. So a triglyceride fat is 3 of something plus glycerol. If you look there, it says it's made from 1 glycerol molecule and 3 fatty acids. So I've got a couple of pictures so that you can see kind of what it would look like drawn out. Again, I don't need you to memorize the formula, the structure, but just to recognize its general appearance. This part here is your glycerol. It's often called the backbone because it's sort of what holds the whole thing together. You could imagine it as being sort of like the spine, and then each of the fatty acids is like a nerve sticking out of the spine. So that's glycerol, and these long chains are fatty acids. So you'll see they're made up of carbons and hydrogens attached together. Now I'm just going to make this a little bit bigger so everybody can see. We have 2 kinds of fatty acids. We have fatty acids that are unsaturated and ones that are saturated. So on the top picture, there are 3 fatty acids. The 2 on the top would be called saturated. But the third one, the one that kind of bends, is an unsaturated fatty acid. An unsaturated fatty acid has, and I'm just going to scratch out the word only. I used to make you distinguish between mono and polyunsaturated, and I dropped that out of there. An unsaturated fatty acid have a double bond. What that means is that right here, between those two carbons, those two letter C's, instead of one line, there are two. That's a double bond. When that happens, you can see that that fatty acid is kind of bent, so it's not perfectly straight across. That ends up creating a softer substance. So the saturated ones, the ones that don't have a double bond and are straight across, they're usually solid, they're harder. Now I mentioned phospholipids, and I'll just remind you, that is what your cell membrane is made out of. Phosphates and lipids attach to each other. And then I also wanted to remind you that I could do dehydration synthesis here to put a triglyceride fat together. I would take a glycerol and 3 fatty acids, and I want you to notice that there are 3 drops of water, so 3 molecules of water, that are removed, 1 for each fatty acid. So essentially, for every bond I want to form, I have to take out 1 water.